A very good day to you all. My name is Anjala Jiji and I'm from Department of Biological Sciences, Arkansas State University. And my mentor is Dr. Asela, and today I'm going to talk about assessing the performance of lightweight aligner for plant transcriptome. As technology has been advancing day by day, methods of analyzing data has also been improved. Traditional analysis, which takes hours to complete, can be done within minutes using these newer platforms. But this analysis has only been performed in humans and other model animals, but has not been done in plants. So my goal is to use platforms such as RStudios to compare the results between traditional and novel technology. And if the results are similar, then these newer softwares can be used in further analysis, which can save a lot of computational time. RNA sequencing is a technology where we extract RNA from the cells, fragment this RNA, convert it into cDNA, and then send it for sequencing. This sequence can be further used for aligning short read to references, count the number of short read aligned, and then do a differential gene expression analysis. One of the alignment tool that I'm going to use for this experiment is called Bowtie2. Bowtie2 is a memory efficient tool for aligning and sequencing. One of the main benefits of Bowtie is that it is more tolerant to low quality bases. But as you can see on your right, the time used for Bowtie analysis is too much compared to all the other softwares. The second alignment tool I'll be using is Callisto. Callisto is a mapping program. It creates some sort of approximation of the reference and read rather than actually matching it to the sequence. As you can see, this is a comparison between time and accuracy. In the first graph, we can see the time used by Callisto and time used by Bowtie has a huge difference. Where Callisto just uses 5 minutes, Bowtie takes almost 1000 minutes to do the same sequencing. And when we are comparing the accuracy, we can see Callisto and Bowtie have almost the same number in accuracy. Why are we doing this in plants? Plants have a large variation in their genome size and their level of ploidy. This diverse and complex nature of plants make their genome very difficult to analyze. And Callisto has never been tested in polyploidy plants, but have recently been done with diploid plants. So a very systematic study of this genome is important, and these tools are important so that we can save time and other resources in future. The goal of my project is to use diploid crop plant samples to compare different alignment methods to reduce the analysis time and to develop an analytical pipeline for this crop for future references. And the main objective is to compare the performance of Callisto and Bowtie2 in Arabidopsis sample. The data for this experiment was obtained from a paper which originally had 17 samples, but due to time restraints, we could only do 10 samples. The methods I will be using is use the reference and map it using Callisto and Bowtie2, and then do an abundant estimate using Callisto and RSIM, and then do a differential gene expression and assess performance using RStudios. After analyzing both the softwares, we got a volcano plot and a PCA plot. As you can see in figure 8A and 8C, both the plots look exactly similar with very, very minute changes. As in the case of 8B and 8D, we can see all the groups were almost group similar. So from this plot, we can see that there is no much difference between the use of Callisto and Bowtie2. From this Venn diagram, we can see that majority of the area has been overlapped between the differentially expressed gene. There is a very slight difference or very slight area which there is no overlap, and it can be majorly because of some false positive because they are from the same experiment. This is a plot showing the count between bowtie 2 and kallisto. As you can see, the red one represents the bowtie and the blue one represents the kallisto. And we can see the difference remain consistent throughout. I think kallisto stands out because the repetitions may have been counted while bowtie didn't count it. From the results, we can see that there is no much difference between Callisto and Botai's analysis. But when we did this experiment, Callisto took more just 3 hours, where Botai took more than 2 days. So we can conclude that Callisto is ultra fast and almost gave us same accurate result as Botai. And in future, this lightweight and fewer options will make the everyday analysis so much more easier. And we can see from the initial analysis that it can be used in non-human or mouse samples. And Callisto and all the other newer technologies can change the way we do data analysis. And these are the citations of the paper that I used for this project. Thank you and have a great day.